Okay, so we're finally going to talk about the science behind genetic engineering at this point. So, the science is really very common lecture material that you would have seen in our forensics course if you have taken this class in our program at this point. If you've not, no big deal. I'm going to still cover the basics here. And if you have taken that class, well, guess what? You get to sit in with a review. And maybe that's good for you because you might not remember what you need to know about DNA for this lecture. All right, so a little bit about DNA. First, DNA stands for deoxyribonucleic acid, so that is how you spell it. You probably know that's going to be a test question. I mean, all of this is driven by DNA, so you know it's going to show up, and it's science related as well. So, deoxyribonucleic acid. The ribo part is because we're talking about ribose, and ribose is a sugar or a carbohydrate. So we've talked about sugars and we've talked about carbohydrates at the beginning of the semester. This is a sugar-based structure that has some modification on it, and then this is enveloped into a larger, bigger picture of the puzzle. So DNA, that's where it starts. It's found in all organisms, and it's also found in all plants. In addition to that, DNA is what we would call the genetic code, and that's really made up of 4 nucleic acids. And those nucleic acids are going to stand for A, T, G, and C. That stands for adenine, thymine, guanine, cytosine. So if you don't know how to spell those, or if you don't know what those stand for, my advice is to go to Wikipedia, type in DNA, and then look for the abbreviations of A, T, G, and C. And they're going to spell them out for you. They'll give you the structures and all of that. However, here, I'm not really concerned with that kind of thing. All right. So these 4, A, T, G, and C, they have some properties that is very common to any organism. The G's and the C's always pair up together. Whenever you see one, you see the other one. And the A's and the T's will always pair up together. When you see one, you'll see the other one. Now, when I say pair, we'll see that in just a second, but this is the concept of having things organized. And it's quite astonishing back in the day that these 4 nucleic acids could give us enough difference to give us the diversity that we saw in just people alone, much less everything else. So everything that's out there, whether it's you or whether that's weed in the corner of your house, or whether it's the grass in front of your yard, or whether it's the bug that's on the grass in front of your yard, all of that is made up of DNA, and all of that is mainly made up of A, T, G's, and C's, just in a different order. That is the only difference here. They pair up in a different order. They start structuring this DNA chain, if that's what you want to call it, in a different way. And then all of a sudden, you have a person on one hand and you have a butterfly on the other one, right? So DNA is typically made up of 2 strands. Here's the first strand, and here's the second strand. These strands are complementary to each other, all right? They do have a direction. We typically call it 3' to 5', so therefore this one would run backward, 5' to 3'. And these are connected, and they are copies of each other, complementary copies of each other, meaning, like I said before, for every A, the other strand would have a T. For every C, the other strand would have a G. And this will constantly repeat down the chain. So I could have another C, maybe a G. So here I'll have a G, and then a G, and then a C, and then maybe I can get a few T's that are sandwiched in here, TTT, and on the other strand, I'll have an AAA. So this is how DNA is going to be organized. These strands can be separated very easily in your body at a controlled physiological pH and at a controlled temperature. This is very stable. It stays around for quite some time. However, if we add some heat, which is represented as a triangle, to DNA, we can get these things to separate from each other. And this is really where the magic can happen in the genetic engineering world. Okay, if we can separate these two strands and have a loose strand here and a loose strand there, and if we give this environment the proper ingredients, what can happen is that it will look at these freed up DNA strands, and it will look at the base pairs that are on that strand, and it will go through and zoop, and then it would start matching the base pairs that's needed to create a brand new DNA double strand for me. The same thing happens over here on this side. So what was one DNA molecule can actually be copied, and I can get two DNA molecules from it. So if I give it the proper ingredients, if I give it the proper reagents, I can amplify the DNA sample that I have in front of me. That requires something that we call primers and what we call Taq polymerase. All right, Taq polymerase is going to be an enzyme, and it's the enzyme that does the work. This is the enzyme that puts it together, matching all of those base pairs, one after another, after another, after another. The primers are going to be areas where we can start the replication. That's what those are, right? So as you can imagine, human DNA is about 6 foot in total length from one end to the next if we stretch it out. Where in that 6 foot molecule do we begin the replication process? Well, the primers will go in and it will lock into place an area that it wants to start. So Taq polymerase will come in and it will ignore the 6 foot chain, and it looks for the primer and it says this is where they want me to be. So Taq polymerase sits down onto the chain and it begins to grab a hold of it, and then it begins to pull it through. And as it's pulling the chain through, it's pairing the base pairs up with it one after another after another. Okay? Primers, there's tons of different primers that are out there. Primers are made in such a way so that you can target different areas of the DNA chain. As you would expect, not everyone is focused on the same part of a DNA chain of an organism. So there's different primers that are made, and they are specifically chosen and picked to go to the areas that will be closest to the gene that you would like to take a more interest in, or that you would like to maybe modify, either splice in or splice out. So tons of different primers are out there. There's actually a database of primers now that you can go in, tell it kind of what you're looking for, and it will give you the best primers for you to choose for the research that you're doing. In addition to this, we often will not work with the entire DNA chain at one time. As you can imagine, this is a pretty large molecule, not only for humans, but maybe for some plants as well. Bacteria typically has a little bit of smaller DNA chain associated with it, but we can chop these DNA fragments up, right? So this big long strand, I can go in and I can chop it up into multiple pieces. Again, I have different reagents that can allow me to chop the DNA strand up in different types of pieces, but this gives me a way to analyze the DNA in a little bit different detail and maybe with a different perspective. These fragments that I begin to cleave them up in, the pieces, they can be analyzed, and those are going to be analyzed on something that we call electrophoresis. All right, so the concept here is to take DNA strand naturally in its home environment, rip it apart, copy them, and do that multiple times, over and over and over, until we get quite a bit of DNA. Then I can go in and choose primers that target specific parts of the DNA molecule that I want to know more about, and then I can use Taq polymerase to go in and copy those areas of the DNA. And over time, those pieces or those copies will get copied more and more and more and more. So it is a way that I can take this big old huge piece of DNA and really only focus in on certain areas, briefer areas, shorter areas, and I don't have to deal with all of the other stuff that really is not going to affect the genetic code that I want to change. DNA wraps around on itself. When this begins to wrap around, we call this the helix, and this is a DNA helix. What you're seeing are two strands. You're seeing one strand that goes here. The other strand runs in the opposite direction because they're complementary of each other. The A's and the T's pair together because once they are paired, they give me a certain length, right? And then the G's and the C's are paired together because once they're paired, they give me actually the same length. It doesn't look that way in the diagram, but it is, and that's one of the reasons that they always pair. The number of hydrogen bonds is a driving force, but the length of those bases when they pair up are also another driving force. It keeps the DNA molecule very uniform, all right? And that's really one of the things that we're after. What's up next? Okay, well, we talked about replication, right? So replication is in this field is titled polymerase chain reaction, and this is abbreviated as PCR. So what happens here is my intact DNA strand, 3 to 5, 5 to 3, right there, all the base pairs together. And I'm going to, number one, heat that DNA strand up. And we normally heat it up to 94, 96 degrees. Not quite boiling water, but close to it. All right. So this will separate the strands. I'll have one strand here and I'll have two strands here. And then what we do is our ingredients, or our vegetable soup ingredients, will include primers. And that is the very first step after the DNA separates. So in step number 2, you're sealing what we call an annealing process. That annealing process normally has a range that's associated with it. This diagram is presenting you 68 degrees, but it can actually be a wider range than that. It can go 40 to 60, 40 to 70 degrees, somewhere in there. It really depends on the primer that you're using, and that's why there's not one specific temperature in order to go by. Number 2, after it anneals, what does that actually mean? Well, the primer begins to hook up with the DNA strand, and it kind of locks in place, and that's where it needs to be. So after we give this some time to go on, then we move on to step number 3, and that's the elongation. And the elongation is when the Taq polymerase comes in, and then it begins to extend that area where the primer begins to sit. So notice here's the primer in red, and now we have elongated the DNA chain. The same thing happens down here at the bottom. Here's my red primer, and now we have elongated the DNA chain after that primer. Then this process is repeated. It goes back to step number 1. So the DNA strands are together again, right? A new one formed. They're together. So I need to separate those copies, and then I need to anneal it again, and then I need to replicate it again. And then I go back to number 1 and separate all the copies again, and then repeat this process over and over and over. Normally, this is around 30 to 40 cycles. That's what we call this. And this step is called a cycle. So step 1, step 2, and step 3, the denature, the anneal, and the elongation step together is called one cycle. So this is a way that I can start looking at DNA. It's a way that I can start replicating DNA. It's a way that I can start getting DNA that's maybe a little bit more important to me, and I can leave out all of the unused DNA that I don't really care anything about at all. This used to be done by hand with a bunch of water baths back in the day, but now we have what we call PCR instruments or thermal analyzers. Either one of those are the same term that's regarded to the same thing. The one to the left is what you'll see in our laboratory at the moment. This is a smaller PCR instrument. We don't do a lot with PCR and DNA, actually. We only use this sucker a couple of times. But if I was working in a real genetic engineering lab, or if I was working in a biotechnology lab that dealt a lot with DNA, I would look at something over on the right-hand side as the instrument. This is actually 4 of the ones on the left made together, which is why you see 4 wells that you can load samples up into, and 4 lids on the top. So it will allow you to run 4 different sets of DNA in a thermal analyzer at one time. And I can start one and maybe have the other three still open and vice versa, and just really have some flexibility or some freedom as far as the instrument goes. Of course, those are more expensive, but the option is to buy 4 of the ones to the left, and that would be even more expensive if you did that. Here's the gel electrophoresis scheme. We'll talk a little bit more about this in just a second. But normally what happens here is that when I get the DNA that I want to work with, there's some times that I will have to look and separate those pieces in order to figure out what's actually there and what I've replicated. Or in the genetic modification world, I could also use this to identify certain genes that might be present in genetically modified food. So if someone grew corn and that corn was sold, I could bring that corn into the laboratory, I could smush it up, I could do some PCR amplification to amplify the DNA, and then I can run that DNA on a gel electrophoresis. And I'll look for certain banding. I'll see some bands that show up on my gel, and if particular bands are going to be present, then I can compare that to a band that I know is genetically modified, and then we can label that food item as genetically modified because it shows the gene of interest that we're looking for. So up here at the top are the wells, and what happens is that the DNA sample goes into the wells here, and I'll load those up very carefully. This whole thing sits down into a buffer. That buffer keeps the physiological pH or the pH unchanged over the course of this analysis, which is, again, very important. And there's 2 probes or electrodes on the end. One will be negative and one will be positive. Positive is typically the red color that we always associate with it. And the reason that I'm mentioning that is because the DNA sample, it's very important that I load it closest to the negative side. In doing that, DNA is a negative structure. So this negative electrode will push these negative pieces away, and the rate of migration starts to go this way. In addition to that, down here at the bottom, this positive electrode will create some positive potential down here at the bottom of the well, and then the DNA will begin to migrate closer and closer and closer to the end where those positive charges are. Over time, the smaller pieces are a little bit more agile and they can move a little bit better than the big, chunkier pieces. And what will happen is that I will see a natural separation beginning to take place on my gel, where the smaller fragments typically go to the bottom and the larger fragments typically stay up to the top, right? So what I'm looking for is I'm looking for these particular bands. And does it have a band that is associated with genetically modified food? Okay, this would be something very similar that we would do on a crime scene with a person. If we were trying to compare the DNA samples of people, if we were trying to compare the DNA samples of a child to the mother and the father, this is a very similar process that goes on here. A real picture of a electrophoresis chamber looks like something here on the screen. You see the agarose gel. The agarose is the material that makes up the gel-like stuff here in the middle. You see the wells. The dark blue areas are typically indicators that some kind of sample is loaded up in there. We call it loading dye. Loading dye is typically a darker blue color, and that's a very good sign something is present. The black area here to the left-hand side of the screen, this is your negative area, which is the side closest to the wells. And then over here, this red is going to be your positive face, and my samples will migrate this way. It's very important that you line up your gel the proper way. Sometimes the DNA fragments cannot be seen with the naked eye, and sometimes we need a little bit of help. So when we begin to look at these gels with DNA in them, we're seeing some very fluorescent kind of colors that can come from these sometimes, but very often it needs help of what we would call a light box, a UV light box, or some type of light box dialed in to a wavelength that's specific to the indicator that I have used in my experiment. Some of these indicators are very poisonous. Some of these indicators are mutagenic. So you just want to be careful. I mean, they are interacting with DNA. And if it interacts with DNA, it's going to interact with your DNA too. So it's very important that you wear gloves when you work with these indicators, that you're not messy with the indicators, because this is a source of contamination and it's a health concern as well. So what you're seeing on the screen is an electrophoresis gel to the left, and that is on a light box, which is the bulbs that are behind it, and it's showing the bands up maybe a little bit better for you. So that way you can visualize them much better than with the naked eye. All right. Over here to the right-hand side, this is probably like a computer image of a DNA gel. It shows up the colors a little bit better. It takes some of the background color away, so the bands actually show up much clearer and much better for you. So that way you don't have to question, is that a band or is that not a band? Here's another picture or another image of DNA banding. Again, this just really determines on what kind of indicator that you want. Sometimes these things are blue, sometimes these things are green, sometimes these things are pink and purple. It really just depends, again, on the reagents that you decide to use in your imaging step of the DNA. Here's another image of DNA. I know it doesn't look like a lot, but each one of these bands, they're very clean, but they also represent a different segment of DNA. So the bands up here at the top, these are the larger bands, which mean number of base pairs that are kept together. And then as we cleaved that DNA into those pieces, the pieces are separating here for me, and the smaller pieces are down here at the bottom. So that means smaller, meaning lower number of base pairs that make that fragment up. Okay. Here's another example. It's kind of a textbook workbook example of what we would be looking at if we were analyzing something for genetic modification. So to the left, what you're seeing are a couple of different rows, 1 through 7, and it gives me the key over here to the right-hand side. So lane number 1 is a non-GM food with a plant primer. The plant primer is just used to let me know that this did come from a plant, all right? And we see the plant primer show up right there. And then number 2 says this is a non-GMO food product with a GMO primer. So this primer was looking for very common genetic modified areas of DNA that we know about, and this GMO primer, it didn't see any, right? So therefore this lane number 2 is completely blank. There's nothing there, and that's because genetic modification was not there. The primer did not work for me. All right. Number 4 is a test food. I'll come back to these in just a second. Number 5 is a GMO positive control. So here, number 5, this is a genetic modified food with a plant primer. Well, it was a plant, so I should see the plant primer. It's right there, okay? And then lane number 6 is the GMO food with a GMO primer. And if I scale down through number 6, I see it. It's right there. So this is the primer that is the genetic modification. This is a way that we know what to look for or what they would like for us to look for. Finally, number 7 is what we would call a molecular weight ruler, or something that we call a DNA ladder. And the ladder, its only purpose is to give me an idea of numbers of base pairs. That's it. All right. So for instance, that gene, that genetic modification right there, well, I know the size of the base pair or that fragment because I've ran a ladder, and the purpose of this ladder is to give me a bunch of different bands at a bunch of different lengths, and that way I can kind of compare that piece, that fragment of DNA against it. All right, I get an idea of how large. Number 3 and number 4, I'm going to go back to these because these were the test foods. So number 3 was a test food. I did not know if it was GM'd or not. And I ran number 3 and I saw a plant primer. So that is a very good indication that this is a plant. It's plant material because the plant primer showed me the plant DNA fragment. All right. Number 4, I'm going to key in, run down, look at there. There we go. This was the plant I now know it's a plant because of number 3. So this was the plant that was given to the lab, and we put GMO primer in it, and that primer did recognize a genetically modified area, and this showed up on my gel, and it also showed up on the positive GMO that we knew for sure was GM'd, and it's a match. So this lets me know that that corn or that soybean or whatever they decided to bring in is a genetic modification. Some laboratory genetically modified it to do something different. The one over on the right-hand side, number 3, you see the plant gene, which is good. So that means there was a plant that was brought in. And number 4, you didn't see anything at all. And that was a very good sign. There was no match, because if it did have genetic modification, it would have shown that base fragment right there. Now, don't be surprised. These are textbook examples, and these are very clean, and we could actually see a bunch of other banding that happens in a normal circumstance. What we're looking for really, though, is the gene that has the GM, which is right here. We want to know, are we seeing that? If not, it doesn't matter what else is there. We typically will say this is a negative result for that particular gene that you wanted us to look for. All right. So that's as difficult as these problems get, really. Quite honestly, if I'm looking for genetic modification, this is how we do it. Here's another example. Lane number 1 is that ladder. It's just, again, for base pairs, so I know how big these fragments are going to be. Lane number 2 is a non-GMO marker. So this is a material that has not been genetically modified. 500 BP, which means 500 base pairs, we see the plant chloroplast, which is the plant marker that lets me know this was a plant that was brought into the lab. Lane number 3 was a GMO marker, and this is going to happen at 200 BPs. This is a CMV35S, which is a very common marker that they use in the genetic world. This is the the cauliflower mosaic virus that they use for specific purposes, and I do see a gene that shows up with number 3, around 200 base pairs. Lane number 4 is another GM marker, and this marker is going to focus on another different type of gene, genetic modification, and this is the NOS or NOS terminator. Terminator means it ends, right? So this again is a very common genetically modified piece that shows up in almost every single type of genetic modification as far as food's concerned. So this is another GM area that I can look for, and this is showing up at about 127. Lane number 5 is a GMO food. I know it's a GMO food because as I scroll down, I see the plant chloroplast that's right there that symbolizes it is a plant. And then I come down and there's a match here for the CMV. And then as I come down, I also see another positive for the NOS. So this is confirming that lane number 5, whatever that was a genetic modification. All right. Lane number 6, this is GMO food. And the problem here is that this is a food item and it might not be plant. I'm missing that plant one, right? So this is food, but what type of food? I can't say that this is a vegetable. I can't say that this is a fruit. I can't say that this is something like corn, but it's something that has been genetically modified. And as I go down, then I'm going to see here is a gene marker and there is a gene marker. So this is genetically modified, whatever it is. So that just gives you an idea of how we look at PCR, how we look at electrophoresis in order to determine if something given to me in a laboratory is genetically modified or not. That's the only thing that I'm looking for here in this video, right? So a little bit of background about DNA, separating it, copying it, the ingredients that are needed in order to copy it, and then allowing it to PCR, putting it into an electrophoresis with some restriction enzymes that cleave it up into fragments, and then looking at the fragmentation pattern. And that's what we're seeing here. It's a fragmentation pattern, all right? So in the next video, we're going to talk about how this is accomplished. We're going to go through and talk about genetic engineering on the other side, all right? What would happen if I was told, you've got to genetic engineer this flower? Okay, how would we do that? What I've talked about here is how to detect it. All right. But what if I was working on the other side of the house, which is doing it? So we'll take a look at that technology as well. You're going to see that this is not very hard to do, honestly, and it might look a little bit weird to you when you figure out what we actually do, but it's something that's very commonly done. So of course it's going to be a little bit easier than what you think. Stay tuned.